Hello everyone. This is a molecular biology chapter for a B.Sc course, 6th semester, paper 8, biochemistry. In this session number 38, I am going to explain you the mechanism of DNA replication. I am Dr. Veena K from Maharani's Science College for Women. In this session you have mechanism of DNA replication and role of DNA polymerase in DNA replication. See, the mechanism of DNA replication was first given by Arthur Kornberg, who is a Nobel Prize winner, and other biochemists deduced the steps for this replication. So, which are the steps involved in DNA replication? You already know that DNA replication is by semi-conservative method, and the different steps involved are initiation, elongation, and termination. So here, in the initiation step, the protein binds to DNA and open up double helix, and in the elongation step, protein connect the correct sequence of nucleotide into a continuous new strand of DNA, and in the termination step, protein releases the replication complex. So it starts at the origin. DNA replication starts at the origin. So here you see, I'll show you here, you can see clearly where the DNA replication takes place. It starts at the origin. Okay, this is in the case of prokaryotes and also in the eukaryotes. And remember, this DNA replication is bidirectional in nature and the replication fork moves in the opposite direction. Okay, so please remember, it is bidirectional replication and the replication fork moves in opposite direction. Come on, let us learn now what are the mechanism involved in the semi-conservative replication of DNA in prokaryotes. So DNA replication occurs in the following steps, each catalyzed by an enzyme. Here, your important what you have to notice is each step requires one enzyme. So you should know the name of the enzyme that is involved in each step. See, every DNA molecule has a specific AT-rich region called as origin of replication or ORI, ori site. We call it as a ori site. The enzyme DNA helicase binds to the ori site and brings about the unwinding of the DNA molecule. So, you have learned DNA is a double-stranded structure. So, for replication, it has to unfold. So, that is done by this DNA helicase. Okay. Now, in next step, once the DNA is separated, remember, this separated DNA should not again recoil. Okay, so there should be some enzymes which should prevent this recoiling, and that is done by single-stranded DNA binding proteins or SSP proteins. In the next step, the unwinding of the DNA molecule at one end of the replication fork causes a negative pressure generated at the lower end of the fork. Okay, obviously. So this pressure might break the DNA molecule, hence the enzyme DNA gyrase binds to the lower end of the fork to relieve the negative pressure generated and prevents DNA damage. Coming to the next step. The enzyme DNA polymerase synthesizes a small stretch of RNA molecule at the 3' end of every template strand, and this small stretch of RNA is known as the RNA primer. All this I am going to explain you through the diagram, which will be more clear. Okay. So in the next step, the DNA polymerase recognizes the binding sites to the RNA primer. Okay. So what it does, DNA polymerase specifically recognizes the binding sites to the RNA primers, and all this is required for that. And in next step, on one template DNA strand, synthesis of DNA occurs continuously, that is in 3' to 5' template strand. This is continuously, which is synthesized, is known as the leading strand. In the next step, on the other hand, that is on the other template strand, synthesis of DNA occurs discontinuously. Okay. This strand synthesized discontinuously is called as the lagging strand, and the fragments of DNA synthesized on the lagging strand are called as Okazaki fragments. Now, Now you know what is leading strand and what is a lagging strand. You get a question for 2 marks. What is a leading strand and what is a lagging strand? So please remember these two. In the next step, once the entire length of DNA is replicated, the DNA polymerase carries out the 5' to 3' exonuclease activity to remove and replace the RNA primers with DNA. This is called as the RNase activity of DNA polymerase. In the next step, DNA polymerase also performs 3' to 5' endonuclease activity for removal of wrongly incorporated nucleotides, and this is called as the proofreading activity of DNA polymerase. And the next very important step is all the fragments of DNA synthesized are joined by the enzyme DNA ligase. Okay? So that forms a phosphodiester bond between the DNA fragments, and DNA ligase is also known as the molecular stitches. Like how the tailor stitches the cloth pieces, similarly this DNA ligase stitches the DNA fragments. Look here. What all I explained now, I am going to explain through this. So this is a this is a double stranded DNA. So there are 2 strands. They run in an anti-parallel direction. See, this runs in 5' to 3' direction and this runs in 3' to 5' direction. Now, this DNA helicase, look here, it unwinds. Once it unwinds, these SSB proteins are here to stop this recoiling. Okay, next. In this strand, the new DNA strand complementary to this is synthesized in 5' 3' direction towards the replication fork. So this is called as the replication fork. Whereas in 3' to 5' template, this new strand of DNA is synthesized opposite to that is away from the replication fork in 5' to 3' direction. See here. So here, which is this new template strand is synthesized in a discontinuous manner known as the lagging strand or with Okazaki fragments. Here you can see there is a continuous synthesis. The new template strand, which is known as the leading strand. For that, what are you required? You require RNA primase, DNA primase, and this molecular stitcher. Look here. These fragments are joined by this DNA ligase. So this is the complete mechanism of semi-conservative replication of DNA. Remember, it is semi-discontinuous replication and the new strand synthesis is always in 5' 3' direction. See here, you can see here a leading strand. This is a lagging strand, which are known as Okazaki fragments. 2 DNA strands which are anti-parallel to each other, and these are the new strand, and the new strand is synthesis always in which direction? 5' to 3' direction. So the same thing to make you more clear, I have explained you with the nucleotide basis, how this 3'OHN attaches and how the bases are paired complementary to each other and how a leading strand is formed and how a lagging strand is formed. So, what is the role of DNA polymerase in replication? See, the DNA polymerase performs 3 functions, that is 5' to 3' polymerase activity for the synthesis of new DNA strand, exonuclease activity for removal of RNA primers, and 3' to 5' endonuclease activity for proofreading. Now, what you have to remember and what you have to learn in this session is which are the enzymes required for semi-conservative replication and what is the job of each enzyme, the meaning of leading strand and the lagging strand, and what are Okazaki fragments. Sometimes they'll ask you for complete semi-conservative replication for 4 marks or leading strand or lagging strand replication for 2 marks. Now, see what are the core proteins that are involved in this semi-conservative replication? Topoisomerase, helicases, SSB proteins, primase, DNA polymerase, okay, tethering proteins and DNA ligase. Now, what is the job of each one of these? I have very clearly mentioned here. Topoisomerases prevent torsion by DNA breaks. Helicases separate two strands, and SSB proteins prevent re-annealing of single strand. And what is this primase? Where a group of what I have already explained you in the previous slides. RNA helps in RNA primer synthesis, and DNA polymerase synthesizes a new strand, and tethering of proteins stabilizes polymerase, and DNA ligase seals nick via phosphodiester linkage. That DNA ligase is also known as a molecular stitcher. So this is about the semi-conservative replication of DNA. So you clearly remember and try to write this. Okay, how each one of these helps in DNA replication, how the process starts, and in which direction the leading strand moves, and why it is called as a leading strand and why it is called as the lagging strand. I hope you have understood the mechanism of DNA replication. Thank you.